Hi, I'm Noam Tissier, and I'm a bioinformatics scientist here at Arc Institute. Today I'll be telling you all about a tool I've been developing called Cyto, an ultra-high throughput tool for processing 10x-flex single-cell data. So 10x-flex is an emerging single-cell technology that's particularly useful for high-throughput atlas-scale projects. As of now, however, CellRanger is the only tool available to process it. These projects are easily in the thousands of samples, millions of cells, and potentially trillions of sequencing reads. However, individual samples can easily take hours or days to process, even with powerful compute environments. Cyto is a production-ready alternative for CellRanger for flex processing developed here at Arc. It's a reimagining of the internal workflow of CellRanger, tuned and parallelized specifically around the data type and flexible to new experimental designs. The internal mapping algorithm makes use of O1 k-mer lookups, which both map features as well as demultiplex samples, and is highly configurable to alternative sequencing geometries. We introduce a new intermediate binary format, IBU, which is very bare bones and allows us to provide a modular framework for barcode, UMI correction, and building count matrices. We additionally separate and open source downstream processing, such as barcode filtering and guide assignment, and allow for that tooling to be used outside of Cyto. All of the individual components in Cyto can be run and resourced independently, allowing for very configurable and deterministic runtimes. Cyto additionally builds on our work with BINSEQ, a modern binary format for sequencing data that allows us to scale throughput linearly with the number of threads, avoid the I/O limitations inherent to FASTQ, and significantly reduce our storage requirements. The performance metrics speak for themselves. On a smaller scale dataset of 8 billion records from the public 10x dataset of 8 human tissues, Cyto can finish processing in a total of 13 minutes, almost 17 times faster than the CellRanger runtime, with less than 2.4x memory usage throughout. Of that 13 minutes, only 3 minutes or so are spent mapping, showing an average throughput of about 50 million records per second. Notably, we show that almost 32 better performance with respect to CPU hours. And because Cyto is built to be modular, this provides potential to even further reduce computational requirements on cloud computing systems for massive cost savings and improved runtimes. Finally, Cyto uses less than 5 times less I/O than CellRanger, which both significantly reduces the amount of disk space required before, during, and after the run, and reduces the load that mapping takes on network file systems. Importantly, switching to Cyto from CellRanger doesn't require losing any information or changing your results. We report a greater than 99.85% average concordance with CellRanger, measured via Spearman correlation of matched cells on transcript UMIs, with little variation between cells and across samples. When observing the lower dimensional representations, such as PCA or UMAP, we see that we achieve perfect overlap without any method-specific clustering. Cyto builds on our previously released work of BINSEQ, a modern sequencing format that solves many of the limitations of existing formats like gzip FASTQ. The random access built into BINSEQ allows for massive throughput in Cyto, reaching sustained mapping rates of 50 million reads processed per second. And the block compression and 2-bit encoding allows us to additionally reduce our storage costs for our sequencing data by nearly 40%. The take-home message from us on Cyto is that you can process your flex sequencing data on smaller, cheaper cloud instances or just locally. You can reduce your turnaround time by orders of magnitude and just generally make prohibitive computation feasible. Like all our tooling at Arc, Cyto is available free and open source on GitHub. The preprint is now available on bioRxiv, and thanks for listening.